It is a great pleasure to present our latest work here at the OHBM 2022. My name is Wei. I'm a postdoc at Washington University in St. Louis. The title of my presentation is Shared Neural and Genetic Architectures Between Mental Health and Physical Activity. We already know both mental health and physical activity have been linked to emotion, cognition, and brain correlates. And it is well documented that physical activity can improve various psychiatric conditions. However, it remains unclear whether these two constructs overlap in the neural and genetic architectures. In this study, we sought to test this using brain association and genetic association analysis. For brain associations, we applied canonical correlation analysis on UK Biobank data. We tested for mental health and physical activity separately. For brain features, we focused on the three intrinsic brain networks that have been widely implicated in cognition and emotion. Using the Stanford Fine Atlas, we identified 7 subnetworks from the IC components and extracted the mean amplitudes and the pairwise edges of the subnetworks. In total, we had 28 neuroimaging features. In the CCA model for mental health, we included scores of 7 principal components that explained more than 50% variance in the data of mental health questionnaire. In the model for physical activity, we included features derived from accelerometer recording over 2 weeks. Specifically, we applied a machine learning algorithm and extracted the durations of 5 activity types. We also included the average activity magnitude and the metabolic equivalent of tasks. These metrics were normalized over weekdays, weekends, and all recording days. As a result, there were 21 physical activity measures in total. We conducted 2 types of CC models, the simple and unique ones. The only difference between these 2 types is whether to include mental health or physical activity as a covariate to account for the shared variance between these 2 in brain associations. In our models, we also included covariates including sex, age, in-scanner head motion, and data acquisition time. For genetic associations, we employed summary statistics from independent GWAS studies. To align with the brain association analysis, we focused on the same mental health and physical activity phenotypes and conducted 3 different analyses. First, we applied linkage disequilibrium score regression on the summary stat for mental health and physical activity phenotypes, respectively. We then conducted genomic structural equation modeling to derive a latent factor for mental health phenotypes and to examine genetic correlations of this latent factor with each individual physical activity phenotype. Lastly, we applied a latent causal variable analysis to test whether causal relationships can be inferred between mental health and physical activity phenotypes. In our brain association analysis, we found significant canonical variant pairs for both mental health and physical activity, suggesting the engagement of the default mode, salience, and central executive networks in both mental health and physical activity. These associative effects remained significant in the unique models where the shared variance between mental health and physical activity was adjusted. But the coefficient of canonical correlations in the unique models did decrease in comparison to those in the simple models, suggesting there are some shared variance in the functioning of the large-scale brain networks. More interestingly, we found strikingly similar loading patterns of network amplitudes and edges. Overall, we found higher loadings of network amplitudes in contrast to edges in the first canonical variant pair across all models for mental health and physical activity. In particular, the mean amplitudes of dorsal DMN and left CEN, as well as the connectivity between these 2 subnetworks, appear to be the most relevant brain features in the associations with mental health and physical activity. As for the genetic association analysis, we observed significant correlations among phenotypes of mental health and of physical activity, respectively, except for the correlations of risk-taking with neuroticism and with subjective well-being. In the T-cell models, we derived a latent factor from mental health phenotypes, namely negative affect, and found significant genetic correlations between negative affect and several physical activities. After the adjustment for BMI and sex, only the correlation with sleep duration was significant. However, we did not find any evidence supporting a causal relationship between negative affect and sleep duration, nor between negative affect and any other physical activity phenotypes. In conclusion, our study jointly analyzed the resting state network measures and genetic correlations in a large cohort to test the hypothesis of a shared neurobiological basis of mental health and physical activity. Our findings revealed that multivariate patterns of brain correlates were highly similar between mental health and physical activity, and highlighted genetic correlations between mental health and overall physical activity, moderate activity levels, and sleep duration. Taken together, these findings point towards a neural and genetic mechanism that may subserve the protective influence of physical exercise and sleep on mental health. Finally, I want to thank my supervisor, Dr. Janine Bass-Walsh, and all the co-authors and funding agencies for this work. More details of this study can be found in the medRxiv preprint. If you have any questions, don't hesitate to contact me via email or Twitter. With that, I would like to finish my presentation. Thank you for your attention.